Hello there. This is Abhishek and Shushmita here, and we are going to present the poster on topic exploring biophysical properties of membranes and proteins using fluorescence anisotropy. So let's get started. So G.G. Stokes in 1852 first proposed the concept of fluorescence. He proposed that the absorption wavelength is smaller than the emission wavelength. This is called the Stokes shift. So the fluorescence is the phenomenon where a fluorophore molecule emits photons while returning from its singlet excited state to its ground state. So you can see here that the fluorophore molecule absorbs the blue light photons and it emits the green photons resulting in fluorescence and red photons resulting in phosphorescence. So it does either of the two phenomena. So there are cases where there is no emission of photons, that is called non-radiative decay. So it is due to conversion of energy to vibrational modes in an electronic state or due to collisional quenching. So these are the different applications of fluorescence: molecular imaging, then high throughput screening of ligands, pH sensing, cellular staining, and elucidation of drug action. So fluorescence anisotropy. It is the phenomenon where light emitted by a fluorophore molecule has unequal intensities along different axes of polarization. So here a typical fluorophore molecule has different emissions along the axes. These dotted lines represent the different emissions along different axes. So this is a typical fluorescence anisotropy measurement. So the xenon flash lamp acts as an excitation source. It produces light which passes through the excitation filter, which is basically a monochromator. Then it gets polarized in the polarization filter. It gets reflected by the dichroic mirror and then falls on the fluorophore labeled sample. The sample then emits light which passes through this dichroic mirror. It gets split into two at the beam splitter and it enters into the photomultiplier tube where it gets transduced into electrical signals and as detected as output. So this R is the anisotropy formula. And this I parallel is the intensity observed when the emission polarizer and the excitation polarizer are kept parallel. So this is the mechanism of fluorescence anisotropy. All the molecules exhibit tumbling motion in a solution. So let's take this case. When a small tagged substrate is bound to a big enzyme due to high molecular weight, it rotates slowly. And when the polarized light excites the fluorophore, polarized high intensity emissions are obtained. But when a tagged substrate is alone, due to low molecular weight, it spins fast, and when excited by a polarized light, depolarized emissions are obtained. So these are the different factors that influence fluorescence anisotropy. So you can see the viscosity, molecular weight, and cholesterol percentage increase leads to an increase in the anisotropy because they reduce the rotational motion. So let us understand the different plots. So this is a membrane drug interaction plot. So in the control, you can see there is high anisotropy, but when the drug is administered, the anisotropy decreases sharply. So this suggests that there is an increase in the rotational diffusion and an increase in the membrane fluidity. This is this is a plot where there is different phospholipids at different temperatures. So the DPPC at 37 degrees Celsius has the highest anisotropy, followed by the DPPC at 45 degrees Celsius, DOPC and POPC at 45 degrees Celsius. So this is because the DPPC at 37 degrees Celsius uh, is in gel phase, that is, it has high membrane rigidity. And this is because the current temperature, that is 37 degrees Celsius, is below its transition temperature, that is 41 degrees Celsius. While in the other cases, their current temperature, that is 45 degree, is higher than their transition temperature, for which they have a low anisotropy value. So here you can also see that the increase in cholesterol levels is increasing their anisotropy value. So this suggests that addition of cholesterol can reduce the rotational diffusion and hence increase membrane rigidity. So this is a DNA protein interaction plot. So you can see that the increase in protein concentration is actually increasing the molecular weight and hence reducing the rotational diffusion and assisting in the protein and DNA binding. So this is the protein folding kinetics, which is studied under different concentration of guanidine hydrochloride, which is the protein denaturant. So you can see that the anisotropy value increases upon decrease in the concentration of guanidine hydrochloride. So there is a decrease in the rotational diffusion and hence an increase in the protein folding. So the conclusion is that this technique promises to create new avenues in the field of medical diagnosis and research. So these are the references, and thank you.